So we've had our customary 5 minutes, and I thought we should now get started on this. So welcome to the Ian Howard Memorial Lecture. I'm very pleased that Tony Howard is here to see us as well. So welcome, Tony. So it's a great pleasure to introduce the other Tony Movshon over here. And I just wanted to say a few words because that's what I always do. I'm Lawrence Harris. I'm the director of the Centre for Vision Research, which is organising these talks. So Tony and I were actually graduate students together. He was a few years ahead of me, but we were both together in Colin Blakemore's lab in Cambridge back in the 70s. So I do need to tell you a few little stories back in the 70s. Now, they all relate to the fact that it was the 70s. That means that, well, not that sort of really part of it, but the technology wasn't really there. Right? We didn't have computers, at least not personal computers, so everything had to be done by typing. Letters had to be done by typing. And of course, we didn't really have any letters to write anyway as graduate students, but somehow Tony did. He was often to be seen dictating letters to Colin Blakemore's personal secretary. Probably Colin didn't even know, I don't know. And we never found out what these letters were for. But I thought it was very clever of him to, actually be taking dictation as a mere graduate student. The other thing was you could smoke in those days. And you could always tell how long the experiments were going on for, because on the top of the filing cabinet, there was a little row of cigarettes that Tony would be smoking there, and he'd lay them all out, so there were like little chimneys along the top there, so you could see how long the experiment had been going for. The other thing was, of course, we did not have PDFs or anything like that, so we had to write out reprint requests, and people would send us notes and things like that to be able to send copies of our publications. Tony had quite a lot of publications, not quite a lot of these, but nobody could spell his name right. So, in fact, I think on one of the mailings that I sent out, I got it wrong as well. It's a tricky name to remember. And so Tony used to put the stickers from the envelopes all down the side of the door. So we got to see all the varieties of spelling. And of course, he became known as, in fact, Moonshine. So that was one of the ones.
That was Christina's, wasn't it?
It probably was, yes. But I think she got it from that fruit. Okay, but I should say some serious words about him as well. His work has been cited over 24,000 times, and he's been awarded the Antonio Champillemaud Vision Award in 2010, the Carl Lashley Award of the American Philosophical Society in 2013, and the Golden Brain Award from the Minerva Foundation in 2013. He's a senior fellow of the Simons Society of Fellows, and a non-resident fellow of the Salk Institute for Biological Studies. But he's based in New York, at New York University, where he's been since he was a graduate student, smoking and leaving cigarettes all over the filing cabinets. Tony.
Scott, so I didn't have to intervene. It's a pleasure to be here. Good to see old friends. Moonshine, by the way, was a Donald Mitchell version of my name. So I'm not sure where he got it from, but it's inevitably linked in my mind with Donald. It's a pleasure to be here. It's a particular pleasure to be here with Tony to give the lecture in honor of Ian Howard. You all know Ian much better than I did, and you remember his contributions. He was a crucial figure in vision and visual perception for a long time. This book, Human Spatial Orientation, was on my shelf as a graduate student, and it was a sort of very influential book at the time. And then, of course, his later books with Brian Rogers and others on stereo are still sort of archival sources of information of all kinds. And he was a terrific figure, and it's a particular honor to give the lecture in his honor and in front of Tony, who I hope will forgive me for what follows. What follows starts from a fairly simple proposition, which is this, which is that if you have a set of images, or even one image, the task of vision is often said to be to extract the objects from the images. But we know perfectly well that the visual system can't do that job in one step. There's no way to go directly from either pixels or, let's say, photoreceptor signals to objects. And so we know, both from computational and from biological study, that the process is done in a series of steps. And so we've been interested in understanding a few of these steps, in fact, some of the earlier steps in this chain. And before I tell you about the things that we've done, I think I should do what many computational neuroscientists do sort of by reflex, which is to invoke the shade of another great scholar, David Marr, who I knew, and I think Lawrence perhaps knew also as a younger scientist. So Marr, as many of us know, made a lot of contributions to computer vision. He had some seminal thoughts on the organization of visual perception in the visual world and how the brain or seeing machines could break it down. But he also left us with a set of principles that I think are very important to bear in mind when we work as neuroscientists, or indeed as scientists trying to understand information processing machines. And those are these three principles, which are outlined here under the title of levels of investigation, meaning that when you start to try and understand a system that processes information, it's worth breaking the problem of understanding into several different levels. And Marr advocated these three. You can make up others, but these three capture most of what I think is important. First, you want to establish at some level what the computational theory is for the problem you're trying to solve. What's the goal? Why is it appropriate? And what's the logic of the strategy by which it can be carried out? So I might say, I want to locate and identify all of the individuals in this room. I have to individuate them. I have to decide where they sit in depth. I have to decide where the benches begin and the people end, and so on and so forth. And I can sort of invoke that as a general problem, and then I can identify a number of computational strategies for how I might deal with the problem. Now, having identified the problem, I then have to devise algorithms of some kind to solve it. So in the case of the people in the room, I might take advantage of the fact that I have two eyes horizontally separated in the head. They have slightly different views of the world. I can measure the differences between those views and use those differences to deduce the three-dimensional structure of the objects in the field of view, or many other similar things. And then the final problem for any of these things is to settle on the issue of representation and to try and understand at what level of implementation detail is the problem really solved by the piece of hardware that you're studying. Now, that piece of hardware can be a brain, or it can be a computer, or it could be any of a number of other things. In neuroscience today, we're very much concerned with hardware implementation. We've been given in the last 10 or 15 years a whole bucket full of useful tools for pulling apart brain circuits and looking at them in exquisite detail and understanding the computations that neurons perform and the different bits of neurons and how they do those computations. The problem that we face as a field is that, in Marr's view, you should tackle the problems in the order I've given you. That is, you start with the computational theory, then you go to the representation and algorithm, and only then do you worry about the hardware. But there's a lot of neuroscience which is now trying to solve the problem from the bottom up, starting with the hardware, the implementation details, and trying to infer the algorithms and the computational problems. And I think that's a very difficult challenge. It's certainly too hard for me. And so most of what I do, I frame in terms of Marr's levels, and most of what I'll talk about today is really, I think, algorithmic. I'm less interested in implementation detail. I do care whether it's done by a piece of brain or a piece of computer, but beyond that, I'm more interested in understanding the principles of the operations that underlie the problem we're trying to solve. Now, having said that, there is this implementation thing, which is the visual brain of non-human primates, the visual brain of primates more generally, in which these problems are in fact solved by hardware whose nature is to some degree known and understood. So this is a lateral view of the brain of a macaque monkey, more or less as you might see it in the head. And here is a flattened, unfolded view of the same brain as prepared by David Van Essen and his colleagues more than 20 years ago. It's simply made by peeling the cerebral cortex off the surface of this brain and laying it out flat. You see the retinas, the thalamic projections, the primary visual cortex, and then on the surface of the cortex you see about 36 areas marked out here in color, which are known to have visual functions. In the macaque brain, they're about half of the surface area of the cortex. And there is a whole variety of functions that we attribute routinely to different areas in this network. In fact, many of us have been in the business for a long time of trying to decide what the functions are of different areas and pathways through this sort of complicated set of highways and byways. But one of the most important and simplest ways that physiologists have tried to understand this network has been by taking measurements with microelectrodes like this, placed usually randomly close to cells, and trying to understand how the activation patterns of individual nerve cells at different levels can inform us about what the processing job of different areas in the pathway is. So for example, if you do this in the retina, what you discover is that individual retinal neurons have visual responses which are governed by receptive fields that are quite simple in their structure. They tend to be circularly symmetric. They have the classic center-surround organization described by pioneers like Hardline and Kuffler and Barlow. They respond to very large numbers of different objects and elements, and they convey very generalized kinds of information into the CNS. And as we move further into the CNS, the first stop we reach, and the one that was described exquisitely and extensively by Hubel and Wiesel and many people who followed them, is primary visual cortex V1. And I've represented in this sort of cartoon form the receptive fields of neurons in V1 to remind us that individual cells in V1 are selective for patterns of particular orientation and scale, represented here by receptive fields of different tilt and different size. So each one of these neurons signals the presence in the visual environment of spatial frequency components of particular orientation and scale, and sort of breaks the image down into a combined representation of spatial location and spatial frequency. And people think about this, we'll be thinking about this in the course of this talk, as a sort of fundamental representation on which we can build more complex, or at least more sophisticated, sort of signals about images. What we often don't recognize is that the next stop in visual neuroscience, made almost at exactly the same time as Hubel and Wiesel were studying primary visual cortex, was the inferior temporal visual cortex, which was studied in the 1960s by Charlie Gross and his colleagues. And in IT cortex, when you record from single neurons, you find that the simple stimuli that activate cells in V1 and in the retina, for the most part, don't work very well. What happens instead is that individual neurons are often selectively activated by images of objects, complex objects. Remember that transition from pixels to objects? Well, IT seems to be at the object end of that continuum. So an individual IT cell might respond well to the basketball, the pretzel, the pear, and the flag. The particulars of how IT cells respond are still not well understood, and the particular combinations of images that drive any IT cell are also not particularly well understood. And we were interested, and I've been interested for some time, in the question of how we can reason about the transitions that take place between primary visual cortical representations and representations in areas like IT. And so that's led me to concentrate in the recent past on areas like V2 and V4, sort of areas that are not so far up as to be impossible to understand, but maybe within reach of some of the techniques that we've used over the years to try and deal with processing by neurons in these different visual areas. Now, in thinking about how this sequence of information processing takes place, I'm going to be using several different sort of simplifications. One is this pathway is hierarchical. It's commonplace these days to remind everyone that the pathway is both feed forward and feed back, and that areas throughout the pathway, except the retina, send signals both forward and backward. But the fact is that for many purposes we know that a direct single feed forward pathway accounts for a great deal of the variation in response, certainly in the first few tens and hundreds of milliseconds of any particular image presentation. That is to say, basically for most of the time that your eyes rest between saccades. And so we're going to be thinking about this pathway in a hierarchical way, not necessarily purely hierarchical, but as a pathway which elaborates successive representations. And we're going to be thinking about it, and I'm going to think about it with a general framework of successive areas doing as much as we can imagine possible similar things with their inputs, and differing in their properties because they receive different inputs. So the idea is to work out some kind of theme that explains how we can make multiple steps between this representation and this and these, and end up at the complex representation in IT cortex. Now, one of the things to think about in the world, and one of the things to think about in this transition from pixels to objects, is the fact that the world contains more than just objects. And this is this is what passes for a natural scene in New York. This is a cafe in Brooklyn somewhere. This image, like many images of scenes either natural or man-made, contains not just objects, which you might think of as things. We'll call them things, following Ted Adelson, who wrote a nice paper about this problem about 15 years ago. But it also contains reaches of what are best referred to as stuff. All right? That is to say, the textures and continuous elements that fill in the gaps and fill in the space between things. And the visual scenes, all visual scenes, consist of a mixture of things and stuff. And although the words sound silly, the concept is not a silly one, it's a fundamental one, because it represents the fact that visual signals arise equally from elements that are actually pieces of objects, and also from things that fill in the space between. So if I look at the carpet is a texture, and the texture is a sort of self-similar representation, which is certainly an effective visual stimulus, but isn't in any meaningful sense or any usual sense a thing or an object. And so one of the things that we're going to be doing in the course of this talk is asking the question of how much, especially in the early visual pathway, is it even really useful thinking about the properties of objects, and how much are we better off thinking about abstract visual signals, like visual signals that arise sort of from images of stuff. Now, the best way to start understanding the visual pathway remains, perhaps sadly, but remains Hubel and Wiesel's work from the 1960s on orientation, selectivity, and V1. And this is sort of standard textbook stuff. We've all seen this figure, or most of us have seen this figure, showing the responses of a single V1 cell in a monkey to oriented stimuli, showing that the cell responded best to this orientation, less well to others. These images here show the maps of the visual response fields, of the receptive fields that Hubel and Wiesel made. These maps represent regions of excitation and suppression, where light either enhances or suppresses the firing of the cell. They are precisely well thought of as spatial filters, that is to say, templates which can be placed on a visual image, and to the degree that the content of the visual image under the template matches the template, the cell will respond. To the degree that the visual image doesn't, the cell will not respond. And down here is an implementation, a proposed little circuit for how Hubel and Wiesel imagined that orientation selective cortical receptive fields might be built from non-orientation selective inputs from the lateral geniculate nucleus. Now, in a more quantitative era, we tend to study these cells, consider them as filters explicitly, and we often measure them with targets like sinusoidal gratings. By the way, sinusoidal gratings, these things here, are quintessentially stuff, okay? They are definitely not things. Nobody in their right mind would call a sinusoidal grating a thing. And so if you make measurements of the responses of V1 cells with stuff, what you find often is that the cells are responsive to gratings. They respond best to gratings of a particular orientation and of a particular scale or spatial frequency. And you can represent that property by jointly representing orientation and spatial frequency in a polar plot. So 0 is in the middle here. This is just a blob of 0 spatial frequency, which has no orientation. As you move out along a line, you go with constant orientation through increasing spatial frequencies. As you go around an arc, you change orientation, keeping spatial frequency the same. And more than 30 years ago, Russell Develoy and his colleagues measured the responses of V1 cells jointly to variations in orientation and spatial frequency and mapped their receptive fields just by doing what's plotted here, measuring the orientation range and spatial frequency range that would activate the cells. Now, this intuition that V1 cells are joint sensors for orientation and spatial frequency stands alongside Hubel and Wiesel's description of them as measurers of local space and local edges. There's in fact a direct connection between the two. If we make an idealized version of one of these receptive fields by dropping an appropriate function down on this space, and we do an inverse Fourier transform to compute the spatial filter that would produce these tuning curves, this is what that spatial filter looks like, either in perspective view or in plan view. And it's no accident that the shape and size of these regions in this filter very closely resemble the shape and size of the regions that Hubel and Wiesel described in spatial filters. And so this is the account I'm going to work with for the account of what V1 does. What V1 does is it jointly signals space and frequency. It has a sort of four-dimensional representation of these things and passes that on to the visual pathway in its downstream manifestations. Now, the natural question to ask is, what's next? What happens in the immediately following visual area, V2? How does V2 elaborate on the representation that V1 provides? And Hubel and Wiesel, as so often, were the first people to ask that question as well. They studied V2 in the cat. This was in this paper, published in 1965. Area 18 in the cat, in particular, is homologous to monkey V2. This is a paper which you should all read if you haven't. You should read it because it's a wonderful paper. You should also read it because it's the kind of paper that doesn't get written anymore. It's 62 pages long, it has 38 figures, and it begins with the line, "To understand vision in physiological terms is a formidable problem for the biologist." So if you're going to set the frame for something that has large ambitions, that's a good way to go. And so what they discovered was that V2 contained cells that were sort of V1-like in many respects, but also responded rather selectively to particular kinds of elements of the visual scene. In this case, particularly, they found a cell, for example, which responded better to a corner than it did to elongated lines and elongated bars. They made little models, even circuit models, of how the responses of these cells might emerge from convergent inputs from V1, just by extension of the intuition of how the responses of oriented V1 cells might arise from the convergent inputs from the non-oriented cells of the LGN. But there isn't really a very striking difference that they describe. In fact, many of the properties they found in V2, they later found in V1 when they went back to look again. And if you make measurements of the responses of cells to simple targets like gratings, what you find is that V1 and V2 neurons can be seen to have rather similar properties. They respond to the orientation of gratings, they respond to the spatial frequency of gratings, they respond to the size of the stimuli. And measurements of V2 receptive fields based on this idea of sort of elaborating sort of little object elements, okay, have not really taken us very far. There are a couple of examples, I'm putting them up here, I'm not going to go through them in a lot of detail, but these are data from a paper by Ito and Komatsu, these are data from a paper by Anzai and colleagues in Van Essen's lab, showing that cells in V2 do sometimes show responses that suggest that they receive inputs from more than one oriented element. You've probably all seen in introductory psych textbooks, probably older ones, the idea that you make a little cartoon representation of the world by putting little V1 elements around the borders of objects and beginning to draw sort of representations of form. And that idea was first sort of brought up to a higher level by Jack Gallant when he was working in Van Essen's lab. And he had the idea that cells perhaps in V4, in the transition between V1 and IT, might respond to more complex families of objects. And so he tested with a family of stimuli, not quite this one, but a similar one. These are iconic representations, not of individual stimuli, but of families of stimuli that were used in a study by Jay Hegde and David Van Essen a few years later. In addition to the standard stimulus families that I've talked about, like oriented gratings and lines, they showed chevrons and little pluses and spirals and bullseyes and so on. And what's shown down here for a population of V2 cells is the fraction of cells that were most selective in their tuned responses to members of the family shown above. So this says 20% of the cells were most tuned for grating orientation and frequency, another 10% most tuned for lines, which leaves 70% of the cells in V2 better tuned for other stuff, or other things. Maybe I should be careful about using those words. But so unlike the original Gallant study, which I'm not showing, Hegde and Van Essen compared the measurements they made in V2 with V4, where they found this set of data, again showing rather similar prevalence of tuning for all the different properties. But then, perhaps most disturbingly, they did the same experiment in V1 and found very similar distributions. And so we can talk later, if you're interested, about why I think these results are so similar. They have more to do with the way the experiments were done than I think they have to do with the properties of the cells in these areas. But they leave us with this conundrum and this challenge of trying to understand how we should deal with the problem of what V2 is and what V2 does. And so at this point, prodded, I have to say, in large part by some of my colleagues at NYU, notably Eros and Simoncelli, we started to think about the question of whether V2 would show some sort of adaptation for natural images as opposed to just synthetic images like images of gratings and other targets. And that raises the question of what it is that makes an image a natural image and what makes natural images special and different from other kinds of images. So first of all, an image can be anything I make by drawing a pattern of numbers into the display memory of this computer. And most of the things I would generate by drawing numbers into that display memory would not look like scenes from nature. Right? So what is it about scenes from nature that's special? What is it that makes them different and immediately recognizable as natural scenes? Well, there's some low-level truths about natural images which are not by themselves sufficient. So for example, natural images often have a prevalence of lower spatial frequencies, larger elements, but that by itself doesn't identify them. The most salient thing, or one of the most important things about natural images, is that natural images are spatially correlated in an important way. They're correlated in the sense that not just that they contain low spatial frequencies, but that sensor measurements made in different places of the image are highly predictive of one another or highly correlated with one another. So in this image, consider, for example, the fact that these four Hubel and Wiesel type oriented receptive fields that happen to lie along this extended horizon boundary are all going to be co-activated by that natural scene. There's a correlation imposed on the activity of these sensors by the structure of the natural image. They wouldn't normally, by chance, be expected to be activated in that way, but yet here they are. Here's another family which are similar in orientation, different in scale, which are being activated jointly by the sort of the texture that's created by the water. And so what you can say in general for natural images is if you choose any pair of sensors, and you can choose arbitrary pairs of members of sensor families, and ask what the response of this sensor is as a function of the response of that sensor, or vice versa, what you find is that these are strongly correlated. This is simply a representation over a large family of natural images of a particular correlation. And what that means for natural images, not true for synthetic images for the most part, is that if you know something about an image in one place, then you know a good deal about what's likely to be in the image in nearby places. And by nearby, I mean nearby orientations, nearby locations in space, and nearby spatial scales. And those are the three senses in which these things get used. So this knowledge of the statistical structure of images informed some work that Javier Portilla did when he was a postdoc in Sinusoidal's lab about 15 years ago. The task he was working on was actually the task of synthesizing texture. Now this seems a long way from what you might have been thinking about, but the textures that he was synthesizing were actually supposed to be emulations of natural textures. So he would take natural images of things that looked texture-like this honeycomb. And his first attempt was to take measurements with what we'll call a V1 model. That is, imagine taking a whole bunch of receptive fields of different orientation and scale, throwing them down on the image and making measurements with those receptive fields of the content of the image, which gives you a big giant list of numbers. Then you take a piece of Gaussian white noise, which you happen to have lying around, and you throw it down on the screen. And you use the computer iteratively to adjust the white noise so that the measurements it gives you in this domain of V1 sensors is the same. That is to say, what you're actually doing is you're matching the orientation and spatial frequency content of that image with the orientation and spatial frequency content of the natural image. Somebody who knows their way around this will quickly point out that you could achieve the same result just by randomizing the phase of the spatial frequency components. You get the same answer. And what this does is it captures something loosely right about the scale of the image, but clearly doesn't capture what makes it sort of important and interesting. Now, you can then go on to, as Javier did, to build a V2 model. And that V2 model is a model which incorporates information about correlations across position, orientation, and scale. And if you build those correlations in, that's these correlations I was talking about before, and then apply those correlations to this image. So you're constraining the image to match in the space of the V1 model and also in the correlations. You create what I'm going to call naturalistic texture images. And these naturalistic texture images contain the sort of correlations across space from the original natural image. They're not copies of the natural image. And you can make as many of them as you want just by starting with different pieces of Gaussian white noise. So you can make arbitrarily large numbers of these images, which makes them very useful for experiments. Now, in the original paper, so Javier and Arrow showed a number of examples. The top 5 images here are natural images. The bottom 5 are synthetic textures based on those images. And you can see that the synthetic textures work pretty well for the first 4 images. They capture a lot of the basic features of the representation. Richard Feynman doesn't do so well. But Richard Feynman doesn't do well because this is not an algorithm for copying images. This is an algorithm for copying image statistics. And what you're seeing here is a sort of scattered representation of the statistics of the Richard Feynman image. And Jerry Letvin, famous for work he did on the retina earlier, wrote a wonderful paper about this sort of challenge of how to understand texture by just saying that, helping to really make a distinction between things and stuff. What he's saying is that to the extent that visible objects are different and far apart, they're forms, and to the extent that they're similar and congregated, they are a texture. And so these are textures based on these forms. And in particular, this is a Feynman texture based on the Feynman form. Now, having sort of generated these images, the question would arise as to why you would think that V2 would be a good place to look for this signal. And it turns out that there's a very handy signature that V2 offers to help guide you with this. And it may not be immediately obvious why, but this is a diagram showing the sizes of the visual receptive fields of neurons recorded in a bunch of different visual areas by Ricardo Gattas and his colleagues, among others, plotting the size of receptive fields as a function of the eccentricity from the center of gaze, of where the receptive field lies. And in all of the areas, what you can see is that there's an increase in the size of the receptive field with eccentricity. And that increase in size is slowest, neglecting the retina, is slowest in V1, faster in V2, faster in V4. Meaning that V2 cells have receptive fields at any given eccentricity that are about twice the scale of V1. This is a sort of cartoon, what you might be looking at. And so Jeremy Freeman and Arrow took advantage of this to make measurements that suggested very strongly that this texture processing that I'm describing took place in V2. So this is a picture of a bunch of people in Washington Square Park in New York looking at something, I don't even know. So if you will keep your eyes briefly on that red dot, what I'm going to do is exchange this image with another. And that exchange will cause changes, but those changes, as long as you keep your eyes here, will not be perceptible. So I'm just flipping two images back and forth. And if you keep your eyes on the fixation point, I think you won't be able to tell the difference. But if I draw your attention and your gaze over there, I think what you'll see is that the image at that location is being replaced with a piece of what amounts to texture. It's being replaced with a piece of statistically equivalent image. So this guy's face is being replaced by a piece of, think of the Feynman texture. And you're not noticing that. The experiment that Jeremy and Arrow did was to simply vary the scale over which this replacement was done. And they discovered that when that scale was smaller than or at the size of V2 receptive fields, people couldn't tell. And when you made the scale larger, they could. So it was as if the limitation in the human ability to recognize this sort of transformation occurred when the perceptual scale, if you like, was matched to the V2 size. So armed with this information, Jeremy, who was a graduate student in our program, and another graduate student, Corey Zamba, came into my office and said, we want to do this experiment. What we want to do is show these images to V2 cells and understand how their responses work. And so they took 15 different families of stimuli based on 15 different original photographs. 5 of them are shown here. And they took many samples from each, 15. These are the naturalistic textures derived from these images. These are the noise control textures based on those same families. And we simply strung together a movie shown here, 100 milliseconds on, 100 milliseconds off. And we showed this movie to each of the cells we recorded. The receptive fields of the cells were placed in such a way that they were in the center of the image. And the results that you might get were typically of this form. So here are the data I've already shown you, reminding you that V1 and V2 cells have similar responses to, for example, basic parameters of gratings. But when you compare the response of these cells to naturalistic and noise stimuli, naturalistic textures being the ones with the correlations built in V2, there's a substantially enhanced response to the naturalistic texture over the noise, whereas in V1 there's not. And this is one example, and if you look at the population, you can see very clearly. So here's the 100 millisecond time marker for the stimulus. There are actually solid and broken traces here for V1, showing the average over all cells and all textures, showing very little difference between the two families of stimuli. In V2, there's a substantial difference, and the response is enhanced. You can also ask for each cell in V2 what the modulation index is. We measured modulation by simply taking the difference between these two traces divided by their sum, point by point. So that's shown down here. And what you can do is ask for each cell in V1 and V2 what that modulation index is. And here are the data showing a modulation index close to 0 for cells in V1 and shifted radically to the right for cells in V2. So what we have then is a demonstration that V2, but not V1, has this sensitivity to the structure of naturalistic textures in a way that is reminiscent of the way that the visual system might want to go about capturing the sort of correlated features of natural images across space. Now, there are a variety of ways in which one can actually compute this. That we've built models, which I won't take the time to show you, showing you that neural circuits of the kind that you use to compute orientation selectivity from the responses of LGN cells can be built that actually produce this kind of feed-forward signal differentiating the naturalistic textures from the noise textures. So you can simulate these and get a reasonable count of the responses. But there are aspects of this that are clearly not going to be understood in feed-forward terms. So I'm not showing you a feed-forward model. I'll be happy to show it to you later, but in the interest of time, I won't. But what I can show you also is that there's something about this computation which smells of something more complicated. And so a way to find that is to simply look at the responses of cells in V2 as a function of the diameter of these textures. You remember I told you in the original measurement, the textures were always larger than the receptive fields. But so we did experiments to ask, first of all, to ask whether the difference was simply because the receptive fields of cells in V2 are larger than the cells in V1, and that doesn't account for the difference. But we also noticed something quite striking, which is that if you measure the response of a cell in V2 to the size of a naturalistic texture, you might get data that look like this. When you do the same measurement with a noise texture, what happens is you get a measurement that has a much stronger fall off here. So what does this mean? Well, this means that as you enlarge a naturalistic texture beyond the boundaries of the classical receptive field, the area over which the cell sums information, the response is not much suppressed. But when you expand a noise texture over that region, the response is substantially suppressed. And this is true for multiple examples. You can look at many different cells that have similar properties, although the individual curve shapes are different. And so you can plot this modulation index as a function of aperture diameter and show that it rather consistently grows. In fact, if you do the average over the whole population by normalizing everybody so that you scale them by the size of the receptive field at optimum, so what you're plotting here is the relative size of the noise stimulus to the actual size of the receptive field, what you can see is the modulation index grows, and it grows beyond the classical receptive field size. So this suggests that there's some region outside the core of the receptive field that's contributing. You can also ask a question about the dynamics of the response, because the dynamics are often also informative. And by the dynamics, I simply mean the time course of the response of the cell. So this is the average response of 40-some V2 cells just to the aperture whose size matches the size of the classical receptive field, sort of like the optimized aperture. And the traces are all aligned at 0 where the response begins in different cells to overcome the fact that different cells have different latencies. So these are the responses for the classical receptive field stimulus, and these are the responses for the larger stimulus. What you can see is something that's been known before, which is that the effect of the larger stimulus is only felt after the response is about 20 milliseconds gone, which means that for the first part of the response, the surround has no influence, and then later on it kicks in. You do the same thing for the noise stimuli, and again, here's the response to the noise stimulus as a function of its size, and here's the response to the larger noise stimulus, and again, you can see the response kicks in, the suppression kicks in after about 20 milliseconds. What's more interesting is to focus on these two traces and to ask how strongly modulated the trace is as a function of time. That is, how strong the texture signal is as a function of time. And to do that, what we'll do is compute that modulation index for each point in this average trace. And that's shown down here. We're looking at stimuli whose aperture is the size of the classical receptive field. And what you can see is that the modulation signal rises and reaches a peak about 20 milliseconds after the response begins, suggesting that there's a small integration time. Now, if we go out here and measure the same thing for the large stimuli, what we see is that the response now continues to grow for another 20 or 30 milliseconds, meaning that these responses associated with the texture stimulus arise from regions of the receptive field that are larger than the classical center, and they're delayed by about 40 milliseconds with respect to the actual onset of the first spiking responses. And this suggests that there's something more than a simple feed-forward network in play, something that includes a recurrent network perhaps within or between different visual areas. So what I've been talking about up till now have been questions about how these stimuli are encoded by cells in V2. But we were also interested in the question of how the visual system might use the signals from neurons to decode information about these textures. So you could ask a variety of questions about decoding, and we've asked a number of them. And the one that I'm going to share with you now has to do with, in effect, perceptual similarity among texture images. And so, sort of change gears, get the notion of encoding out of your head, and just look at these textures. These are the 5 families of the 15 that I showed you before, and 5 samples from those families shown here. Now, when you look at these images, most of you, I think, will intuitively say these 5 belong to the same family. And they're much more similar to one another than these 5, which belong to different families. So textures that belong to the same family of images clearly have a similarity of structure. And what we wondered was whether the population representation that we could find from cells in V1 and V2 and elsewhere captured these similarities. Now, the way to do that in a population neural representation is to imagine a high-dimensional space within which each axis represents the firing of one neuron, which is encoding some piece of information about the space. So we can start with a three-dimensional example just as a cartoon. Let's say we have 3 neurons, we measure their responses, and these are 2 textures from one family and a 3rd texture from another family. And we might find that in this 3-dimensional space, these 3 textures were laid out so. Now, in order to understand whether perceptual similarity is associated with the representation in this space, we'll adopt the simplifying hypothesis that what matters here is basically neural distance. The things that are closer together, that is, that are more similarly represented by different neuronal populations, will be perceived to be more similar. The things that are further apart will be perceived to be less similar. Now, to try and represent this for you, what we're going to do is actually find us a low-dimensional representation, because 100-dimensional spaces don't lend themselves to presentation in talks. They don't even lend themselves to being thought about. And so what we're going to do is actually project the data down into 2 dimensions using a technique called stochastic neighbor embedding, which was invented by Jeff Hinton and Sam Roweiss about 10 or 15 years ago. It's a technique which is not, for those of you who are into dimensionality reduction like PCA or something like that, it's not a linear projection of the data. It's a projection which is designed to, as much as possible, preserve neighbor-to-neighbor distances from the high-dimensional space when you reduce the data down to the low-dimensional space. So things that are clustered in high dimensions will show up as clustered in this low-dimensional representation. And so we can then ask how the clusters live. So what we can do is take the data from 100 neurons in V1, and we can say, well, how were the 15 different samples of the texture family represented in this color, which happens to be the honeycomb texture family, represented in this reduced dimensionality space in V1? Well, here are the 15 samples. They're kind of scattered. Here's the mean, which is the, the central tendency for all of them. And it looks sort of unpromising, and if we now add the other 14 textures, although it has a certain aesthetics and sort of set this nice, that looks good, but it doesn't actually represent a very effective classification response, in the sense that the different representations of the same texture are scattered widely across the space and scattered in a way that tends to suggest that they would be confused with others. Now, when we do the same mapping in V2, we get a rather different picture. In V2, what you can now see readily is that there are many of these which are clustered, so that clusters of similar colored points mean that in this space, the sort of the reduced dimensionality representation of this high dimensional space, the multiple samples of the textures from a particular family tend to cluster together. That is to say, their perceptual similarity is reflected in the similarity of their representation in this neural space. Now, I can show you completely without content a movie which animates this. It's eye candy, but I kind of like it. So these are the 15 points represented in their V1 space. And if I now animate the transition from V1 to V2, you see all the dots end up clustered together. As I said, it's eye candy, but what's wrong with a little eye candy, right? So to go back to something a little less eye candy-ish, here are the V1 and V2 representations side by side, showing the basically chaotic representation here and the more orderly representation here. Now, those are nice pictures. It's also possible to do this analysis in a more principled way. And we can do it in a more principled way by going back up into the 100-dimensional space and asking how well we can classify different samples and different families from one another using more or less standard machine learning techniques. So we can ask the question in particular, how readily can we tell one sample from the other 14 in a particular family? How well can we tell one family from samples from the other 14 families? And so we can basically put the populations of neurons that we recorded from V1 and V2 to work on 2 tasks. One is family classification, tell the difference between these guys and these guys, column by column, and sample classification, tell the difference between these guys and the other samples in their same families. There are a variety of ways to slice and dice this. We do this analysis, as is common, by choosing different numbers of neurons, but all you really need to know is that all the black points lie below and all the red points lie above the diagonal. What does that mean? The black points mean that for the sample classification, this one, telling different samples apart, V1 performance is always superior to V2 performance for any number of neurons. And for the family classification, this one, the red data, it means that V2 performance is always superior to V1 performance for any family of neurons, for any sample size. So what this means in words is simply that V2 represents the identity of texture families better than V1 does, and V1 represents the identity of texture samples better than V2 does, suggesting that V2 actually is capturing a representation which is more akin to the perceptual representation that we carry around of these textures. Now, I should also add that at this point, what we're seeing in V2 is not a full representation of the perceptual experience we have, and in particular, you can argue quite rightly that naturalistic texture and noise texture, as I've defined them, are just two different kinds of slightly different stuff, and they're not getting us that close to the question of how we represent images and full objects. And so what we want to do now is to ask what happens downstream of V2, and in particular to go to V4. Now, in V4 there are a couple of studies that suggest that this distinction between naturalistic and noise is preserved, and that there is in addition a distinction which we want to amplify in the final study I'm going to tell you about, which is truly between a representation of stuff and things from which they can be made. And so, so we might add now the textures as a question of what job V2 might do. And the only thing I've offered you from V4 is this sort of already admittedly unsatisfactory representation in terms of spirals and bullseyes and chevrons and things. Well, we're going to ignore that. That's not going to work out so well for us. So the question is, what can we say about V4? Well, there are two recent studies about V4 that are suggestive, and I'll use them to frame the beginning of the last experiment. So one was done by Komatsu's lab in Japan, and it actually uses a variation of our texture paradigm. They showed cells in V4, textures and noises made in a way similar to ours. And what they showed was that by some measures, V4 had a stronger texture modulation than V2 did. But by other measures, based on the ability of neurons to discriminate these two families of textures from one another, V4 was in fact not superior to V2. And so my reading from this is that what V4 does with respect to analyzing texture is quantitatively perhaps a little bit different, but not qualitatively different from what V2 does. But something else that V4 does is something that was described a few years ago by Nicole Rust and Jim DiCarlo in a different study, where they asked the following question. They asked cells in V4, and actually also cells in IT, how well these cells could classify pictures of objects or scrambled pictures of objects when the scrambling was done by the same texture algorithm I've been describing. So cells would look at pictures like that, or they would look at the scrambled picture like that, and what Jim and Nicole did was they asked, using another sort of image classification technique, how well V4 could tell the difference between different samples in this collection and different samples in this collection. What they found was, first of all, in IT, that natural objects were much better discriminated than scrambled objects. But even in V4, there was a superiority for natural objects over scrambled objects, meaning that it looked to them like V4 was actually superior in processing images containing things than it was to images containing stuff. And so we decided to sort of shift gears to look in humans and to look using fMRI at this question by asking, in a very simple design experiment, what the representations in extrastriate cortex looked like of these textures I've already described to you, the noise controls, and also the original forms, the original ancestral natural images from which these forms were originally grown. Now, I should say at the outset that there's a sort of a school of thought, I guess, that argues that we should have been studying the responses of cells in the visual pathway with natural images all along, and that this business of reducing the images to their texture sort of sub-equivalence is simply a way of sort of depriving the pathway of its proper input. The problem that we have, and the problem that I've always faced with this, is that when you have any particular natural image, when it falls on any particular receptive field in any particular way, is going to give rise to responses which you can't readily understand unless you survey how the receptive field responds to images in many, many different places, or how the cell responds to many, many different forms of images, many different images that are similar enough in some dimension to be useful, but yet different. So these naturalistic textures are perfect for that kind of experiment because they're self-similar, they have the same statistics, but each is different from the last. Natural images are frankly a pain in the butt for experiments, and I think that natural images used for visual neuroscience experiments with single units are very difficult to interpret. But in the MRI, precisely what makes the MRI less than ideal for some purposes, the fact that you get responses in each voxel that are the average of the responses of thousands to millions of neurons, makes it ideal for this purpose. So we can do this comparison in the MRI and come up with hopefully information that's useful. And the first comparison we wanted to do was really just to replicate the comparison that I've already shown you for V1 and V2, which is what is the response like on average, overall neurons or overall voxels, for noise versus naturalistic textures. So what we have is a very simple block design. We flash naturalistic textures for 9 seconds, and then sort of the spectrally matched noises for 9 seconds. This is a flattened representation of the posterior pole of a human observer, and the early visual areas are identified by a topographic mapping experiment done separately, and these are drawn on here. The shaded regions represent regions that are respectively in sulci and on gyri in the intact brain. And if you measure the response to this exchange, noise versus texture, using this protocol, what you see is something quite striking, which is sort of a direct confirmation of the physiological measurements we made. That is to say, V1 is insensitive to this exchange. V1 doesn't care about the difference between noise and textures, or between naturalistic textures and noise. But all downstream areas, V2, V3, and all the others, are activated by this exchange between naturalistic texture and noise. And so that's a sort of satisfying confirmation of the original physiological finding. It then leads to the question, which is sort of akin to a question which a lot of people have asked in the MRI literature, which is if we take these naturalistic forms, these actual natural images, I should say, and compare them with spectrally matched textures, where do we see strong responses? And it turns out that when you do that comparison of forms versus noise, V1 is again silent, just emphasizing the fact that V1 really doesn't care about much of anything, at least doesn't care strongly about much of anything, except for the spectral content of the images. And yet all downstream areas are strongly activated. You may notice that this area here, which is the lateral occipital complex, is more strongly activated here than it was earlier. I'll show you that directly later. But in some sense, the most interesting comparison is this one, which is what happens if we compare the activation we get for natural images and the naturalistic textures which have the statistics of those images bedded in, but aren't actually the natural images. Think Steinman and the Steinman texture. Feynman and the Feynman texture. Or Steinman. What being at York is what happens. We get your names transposed. The question is what happens here. And the answer is something quite interesting. Again, now when you're exchanging forms for naturalistic textures, not only is V1 silent, but V2 and V3 are for the most part silent. Whereas areas downstream of V3 are actually strongly activated. So this seems to me to be a very direct way of evaluating in a sort of, with some immediacy, this notion of things and stuff, okay? What this says is that if you're exchanging an image of things, this bunch of kids, for an image with a whole collection of stuff of different kinds, then areas from V1 up to V3, which is a fairly advanced visual area, really don't care about that distinction. But downstream, in areas, particularly in the lateral occipital complex, the human homologs of IT cortex, then you actually get a fairly strong and active distinction. And now here you can actually see all three of these MRI images side by side. Here is the texture versus noise image, V1 silent. Forms versus noise, V1 is still silent, and now you can see the stronger activation in effect, the human homolog of posterior IT. And finally, when you do forms versus textures, you see activation which is really confined only to the downstream areas of cortex. So in sort of a summary sense, what you can then say is that we can attribute to V1 the measurement in simple terms of spectral statistics of natural images without particular concern for their organization into objects or object-like elements. Somewhere in the middle here in V2 and V4, neurons acquire the sensitivity to textural statistics. And finally in IT, we actually develop a sensitivity to what you might call form statistics. You could say that vision up here is all stuffish, and vision down here is all thingish. And in terms of MAR, I think what this reminds us is that if we frame the problem of vision and visual neuroscience as a challenge to establish where and how objects are represented, what we can understand is that there is a large chunk of the visual pathway which is indifferent to the question of whether vision is about objects or whether vision is about the elements from which object images are made. And in fact, Lettvin, in that same 1976 paper, has a very nice way of framing it by comparing visions of the visual system with visions of painters of two different schools in the late 19th century. And he contrasts the academicians, painters from the academic school, and the Impressionists, and he says they had mastered ways of painting texture. He said the Impressionists held that texture comes first and form arises from it, while the academicians believed that shape is prior to texture. And he, I've chosen these two examples, but he says the same controversy holds for vision. One can imagine shapes spatially interfering with each other to comprise texture, or else suppose that texture is primitive and textures combine to produce forms, just as letters combine to produce words. It's a sort of an evocative notion, but this makes perfectly clear, and there's, by the way, recent work from a number of people, including a woman called Anne Hermannstadt, who works in John Victor's group, and others, to show that changes in texture statistics across the visual scene are a very effective way of establishing the boundaries of objects. And it's just a way, I think, which is very helpful for thinking about how the visual system sets about parsing the visual world and deciding what pieces of it are actually things, what pieces of it are stuff, and setting about identifying and classifying the things. So let me wrap up by showing you the people who did the work. Jeremy Freeman and Corey Zemba were both students in the lab at the time. Jeremy is now at the Chan Zuckerberg Initiative in California, pushing back the frontiers of some branch of science I don't understand. Corey is a postdoc in the lab still. David Heeger, my faculty colleague and friend, was a collaborator with us on especially the fMRI experiments. Javier Portilla was the postdoc in Eero Simoncelli's lab who did the original naturalistic texture synthesis, and Eero has been my colleague and collaborator throughout all of his work. And so with that, I'll thank you all for listening. Thanks for your attention. Take questions.
Yes, we've got time for some questions. You mentioned earlier on about the down-going pathways, the ones that go back into the, coming from higher centers down, higher inferences, or that sort of thing. How does that relate to the sort of process you were talking about?
Well, so I think one of the things to remember about feedback pathways is that it's fundamentally a mistake to think about them in the same way we think of feed-forward pathways. You can't think of feedback pathways as making spikes in earlier areas. And the reason for that is a simple one if you think it through, which is that if you have receptive fields in V1 that are this big, and receptive fields in V4 that are that big, if there's a strong feedback signal from V4 that makes spikes in V1, then the receptive fields in V1 would be that big. So it's not that the cells in V4 are sending signals back. They are modulatory in nature. They must be altering the responses driven in earlier areas by their afferent feed-forward connections. The connection I wanted to make, I maybe didn't make it strongly enough, is in the data I showed you about the effect of size and time on the responses to the modulation responses to the naturalistic textures. What I showed was that areas larger than the classical receptive field and times delayed from the onset of the response were the ones where the information about naturalistic texture seemed to become manifest. And that suggests to me that areas that contain large visual receptive fields and areas whose responses are slightly delayed in time make a contribution. And so that seems to me to be a place where feedback signals may actually play a role in this. I think, yeah.
First, just a comment, I believe there's evidence that feedback signals are largely NMDA, which would serve to multiply rather than to produce spikes, which I think is consistent with what you're saying. And the second question was the delayed surround facilitation sounds very much like a higher level version of collinear facilitation than a simple V1. Is that sort of the direction you're thinking?
Yeah, I think that's right, and I think that those collinear interactions are probably mediated by similar mechanisms. So, yeah, yes to both.
Just a very basic question, but more Kubo-Weasel type thing. So you showed a picture of like a tonotopic map from Thorell in the early 80s of kind of spatial frequencies and kind of orientations you went through. I was wondering, how is phase encoded there, and how might that kind of propagate further through?
So phase is represented by each of the filters. For a moment I thought you said Bayes. I thought, wait a minute, we're going to get into Bayesian arguments here. That seems early. No. Filters in V1 are, let me try and do this as succinctly as possible. So certainly cells in V1, if you look at the shapes of the receptive fields of Hubel and Wiesel type neurons, or if you look at data on the filter shapes of V1 neurons, all phases are represented in any particular place in space. Occasionally people come to the view that there's a quadrature representation with actually sine and cosine phases hanging around near one another. That is an idea whose favorability seems to fluctuate with time. People like it, they don't like it, they find it, they don't find it. But certainly a diversity of phase exists in the filters that are called simple cell filters. Now, in our correlation measurements, which do engage some correlations, phase is important in naturalistic image structure for the following reason, which is that when you have something like a hard edge, which is a common feature of natural images, what you're really dealing with is a situation where the multiple spatial frequency components of the image are phase aligned at the border. So those phase alignments do show up in the correlation measurements that the Portia-Simoncelli model incorporates. What's interesting to me is, in fact, the phase alignment is less important than it is to have an alignment of the contrast power in different bands. So what do I mean by that? What I mean is that lines and edges in images tend to be associated less with actual perfect phase alignment and more with the fact that there's a concentration of high contrast signals at one place in the image. So you tend to have signals at high contrast in multiple spatial frequency bands that co-occur. And those are the things that actually tend to mark the presence of borders in images more than just pure phase alignments. But the answer is that phase is encoded in the early visual areas, especially in V1. Phase relationships are captured by these correlation measurements. There is a conundrum, which I don't think anyone's ever solved, which is that phase-specific representations have not been found outside V1. And yet we are exquisitely sensitive to relative phase in images. So there is this peculiar problem as to how those signals reach consciousness, but that's beyond my pay grade. So. Yeah.
One has to think of Singer's, what you call the binding theory. How do you think all these elements, let's say, orientation, color, or all of the features, they would be integrated somewhere downstream, Let's say everyone gets inactivated if there is what you call, when you present these, the thing at the top, the forms at the top. I don't know, I'm not even.
So my take on the binding problem, as it's framed usually, is that I don't want to caricature and I don't want to go down a rabbit hole with this, but Singer's argument about the binding problem was that when you had multiple sensors with small receptive fields that were responding to the same object, to different parts of the same object, the fact that they were responding to different elements of the same object could be signaled by a temporal coincidence of their spiking activity. It's certainly possible. One of the things that the stuffish perspective suggests is that the individual firing of early neuronal elements may not be that important for this binding. But there is another way that you can solve the binding problem, which is, to me, I think, a more natural one, which is to remember that as you project information downstream, convergent signals reach larger and larger receptive fields, which can actually explicitly combine the representations of different elements of objects. And so if you think about the DiCarlo and Rust experiment, where you have either the picture or the scrambled picture, and you ask how well cells can classify, for example, pictures from scrambled pictures, what you find is that by the time you reach V4, the information that makes that picture an image of an object is explicitly represented, whereas in earlier levels of the visual pathway it seems not to be. So my intuition is that the way you solve the binding problem is part of the general task of solving the problem of representing objects, and you do that by combining the information that's available about those objects from multiple scales and multiple areas.
So I would be very interested to hear your take on the importance of representational structures versus algorithms. So the second level of MAR that you described is also about transformations that have to take place in order to make something useful. But the experiments that neuroscientists do and that you report are really about representational structures, subdividing 15 families. So how would you help us, doing these experiments to do them more in a way that we know what actually is computed, what kind of transformations are really taking place. Because the filter analogy does not really help us in designing experiments.
So, it's a great question. Recognizing that I didn't have time, I skipped over a section of the talk. I don't think I'll rehearse it now, I'll be happy to show you later, which shows you some modeling work we've done, building circuit models to try and account for the modulation responses of V2 cells based on sort of idealized circuit models connecting V1. So these are algorithmic models based on, as you probably know, at NYU we tend to be fond of relatively simple circuit models based on canonical simple computations, summation, filtering, normalization, a few standard things. It's as if you have you're an electronics shop and you have a set of sort of available neural computational parts. And the question is, can you string them together to make a model that gives you a reasonably good account? And the answer is you can, at least for accounting for these statistical regularities, as I've described them in the V2 responses. You can build models that are, it's really not fair to show you the end, but you can build models that are in the Hubel and Wiesel style, in the sense that they're feed-forward convergence models of the responses of individual cells. And you can ask the models how strongly they modulate in response to the different textures. And so what we did was we built models that were like V2 simple cells and like V2 complex cells. Now, these are algorithmic. The limitation that they have, and the problem that we have at the moment with them, is that they are abstract models not fit to data from individual cells. And so what we have not yet done is devise a method to fit them to individual cells. But my takeaway from this model exercise is that it is possible to actually build models of this kind based on more or less familiar parts and get outcomes that at least match our data. One idea which I kind of find appealing and which can be used in this framework, it actually underpins this model, is the following. Imagine that the job of each cortical area is to take local derivatives over the representation of the preceding cortical area. And it doesn't even have to be cortex. So you start with primary visual cortex, which is taking local derivatives over the LGN. Well, what happens when you take local derivatives over the LGN map is you get simple cells. Right? One-dimensional derivatives give you simple cells. Well, now, when you reach V1, if you look at the data, for example, from Callaway's lab and others, what you realize is that there's actually all four of those dimensions that I mentioned are encoded in the map on the surface of V1. Orientation columns, spatial frequency, clumps, whatever they are, and the spatial map of position. So now imagine taking derivatives across that four-dimensional map. Well, what happens is that you'll pick up neurons that have interesting changes in structure. So changes, for example, in orientation over space would encode curvature for you. Changes in scale over space would encode the edge structures for you, and so on. And so, although it's in some sense a kind of a hand-wavy concept, it's sort of an intriguing idea that by doing these successive operations of derivatives over maps and making new maps for each succeeding area, you can actually achieve many of your computational goals. It's part of this idea of going from pixels to objects in small baby steps, one by one. You've got a lot of baby steps, and that's of course the way that a lot of these deep learning rules work. Same kind of principle, same kind of idea.
Okay, so let me remind everybody that we're going to have a reception now over in BSB, in the Behavioural Sciences Building, 164, the regular place where we have our talks normally. And also, we have a little presentation here for Tony. Swag. Swag, yes, nice certificate here to say that you have indeed given the Ian Howard lecture. So, thank you very much.